Hello, everyone. I'm happy to present the work we have and will be doing in a desmoid tumor model in the frog Xenopus tropicalis. So, two models we use the first model is an APC mutant model, where we make mosaic mutant animals by introducing APC guide RNAs together with Cas9 recombinant protein in early embryos. This results in mosaic mutant animals in which desmoid tumors develop with short latency. A second model uses CRISPR-based editors, where we introduce gain-of-function mutations, so missense mutations in the beta-catenin gene. And both models have short latency and a high penetrance. Now, the APC CRISPR model is used in our dependency mapping. And what we do is actually we perform multiplexing by introduction of guide RNAs for APC together with a guide RNA for a candidate dependency factor. And actually, in the tumors that are induced, these are sampled and sequenced, and the targeted genes are, we perform amplicon deep sequencing, and actually we predict that for a dependency gene, we will never be able to sample B allelic frameshift mutations in such a gene. Right? In practice, it looks like this. So we inject APC and gene X guide RNA. For ADAM12, we sample tumors that next to B allelic frameshift mutations in APC also have B allelic frameshift mutations in ADAM12. In contrast, for EZH2, we never sample tumors that have B allelic frameshift mutations. So there's always one allele that is either wild type or that is actually as an in-frame indel mutation. And another gene which I identified like this was PRO3L1. Now, EZH2 is a component of the polycomb repressive complex 2. So we wondered whether SUS12, which is another component of PRC2, is also a dependency factor, and indeed that turned out to be the case. Also here we were never able to sample tumors with B allelic frameshift mutations in SUS12. Now, EZH2, there is an inhibitor available, tazemetostat, and we could show that addition of tazemetostat to animals that carry desmoid tumors, either crispins or APC heterozygous animals, in both cases, tazemetostat in a 3-week treatment of tazemetostat induced a shrinkage of the tumors. Now, to get a bit more insight in how tazemetostat is exerting its activity, we treated human desmoid tumor cell lines, T229, 219 cells with tazemetostat, and performed RT-PCR analysis, and we could show that AXIN2, which is a well-known Wnt response gene, was downregulated significantly by tazemetostat treatment. So what we assume, our working hypothesis, is that strong Wnt signaling activity by APC mutation or beta-catenin mutation requires the activity of PRC2 complex. Right? Tazemetostat inhibits this activity, thereby dampening Wnt signaling responses, leading to shrinkage of tumors. And this brings us to the new project where we want to investigate the mechanisms and the tumor host interactions that mediate desmoid tumor regression by EZH2 inhibition. This is based on the following key observations. First of all, the spontaneous regressions which are observed for desmoid tumors in patients. Right? Now, we know that tazemetostat only is effective in vivo, not in cell culture. Tazemetostat dampens the Wnt signaling strength, and finally, Wnt signaling has been associated recently with so-called immunologically cold phenotype in solid tumors. So this brings us to the central hypothesis that the dampening of Wnt signaling strength by tazemetostat causes desmoid tumors tumor regression via the induction of natural immunity. So immune escape mechanisms have been associated with Wnt pathway activation in solid tumors recently. And mechanistically, this has been associated with reduction of expression of chemokines, chemoattractive chemokines that recruit dendritic cells or T cells, as well as by upregulation by Wnt pathway activation of PD-L1, thereby inducing immune checkpoints. So how do we want to investigate these models? So first of all, we want to use a RAC2 GFP line that we have. Embryos derived from this line will be injected with APC or beta-catenin CRISPR reagents. In the resulting animals, we will investigate whether tazemetostat treatment leads to recruitment of the T cells, or the fluorescent T cells, to the tumor. Right? Of course, anti-immune responses can also be mediated by other immune cells like natural killer cells. However, we lack antibodies to be used in xenopus. Now, we can circumvent this problem by now performing tissue dissociation of the tumors before and after tazemetostat treatment and subjecting these tumor cells to single-cell RNA sequencing, thereby allowing identification of subpopulations of immune cells or stromal cells in these tumors. We also want to analyze whether tazemetostat activity has an effect in immunocompromised animals. And to look at the involvement of T cells, we can use a RAC2 knockout frog that we generated, and we know they lack mature cytotoxic T cells. This was shown by allografting of tumor cells derived from a p53 knockout animal, which we could demonstrate engraftment of these cells in a RAC2 knockout animal. So what we will do now is take a RAC2 heterozygous animal, cross it to a RAC2 homozygous mutant animal, and in the embryos we inject again APC beta-catenin CRISPR reagents. And then, while we expect that in the heterozygous subpopulation we can still observe tazemetostat responses leading to tumor regression. If this tumor regression involves cytotoxic T cells, we expect that tumor shrinkage will not occur by tazemetostat treatment in RAC2 knockout animals. Now we also want to look at these immune checkpoints. Best known ones are CTLA4, PD-1, normally expressed on T cells, and the ligands for them are expressed on the cancer cells. Now, unfortunately, we cannot use these blocking antibodies, which are working in humans or in mice. However, we can easily make knockouts, right? So what we want to do here is we take RAC2 GFP line again, and the embryos are injected with guide RNAs for APC together with guide RNAs for PD-1 and CTLA4. And what we expect to see or to investigate is while recruitment of the immune cells requires tazemetostat in wild-type animals, in a PD-1, CTLA4 knockout, mosaic knockout, actually this recruitment can occur in absence of tazemetostat and already lead to shrinkage of the tumor. So in practice, we may see less tumors or smaller tumors developing in these animals. So finally, I want to reach out to the community. What will be critical for our experiments is the ability to dissociate these desmoid tumors. It's probably not going to be that trivial because of the whole collagen matrix which is present in these tumors. So protocols would be greatly appreciated. We can already explant the tumors, as we showed recently, but this of course takes a couple of weeks, in which time these immune cells will no longer be present. And finally, I want to thank the people involved in this work. Thomas Naar, Dieter Tulkes, Marjolein Caron, and Susan de Monk have been involved in this work. I want to thank the collaborators. And of course, I want to thank the Desmoid Tumor Research Foundation for the support and you for your attendance.